And welcome to our video lecture on topic D2.2 Gene Expression. This is additional higher level content only, so SL friends, you will not be assessed against this content, but it's pretty interesting. You are welcome to keep listening. Our guiding questions for today: how is gene expression changed in a cell, and how can patterns of gene expression be conserved through inheritance? And our objectives for today, we're going to do some vocabulary review. We're going to talk about the regulation of transcription through DNA binding proteins, data boxes, enhancers, and silencers. We're going to take a super quick look at operons, and then also chat briefly about how hormones can affect transcription. We're going to look at the regulation of translation through different rates of messenger RNA degradation. We're also going to take a look at epigenesis, which is super cool, my friends. We're going to look at methylation, acetylation, and phosphorylation of DNA and of the histone proteins that our DNA wraps around. We're going to look at how some of those epigenetic tags, those methyl and acetyl groups, can be inherited and the environmental impacts on those different epigenetic tags. And as promised, a quick review of some vocabulary. So genotype is our genetic information. We can talk about the genotype in terms of like heterozygous for gene A, or I have type B blood. We can also talk about the genome as being the sum of all of our genetic information. What's pretty cool about the genome is that it leads to our phenotypes, the characteristics of us, not just our structures, but also our functions. So curly hair, but also the ability to make lactase and break down lactose. It's our phenotypes. These are definitely related to genotype, but also to our environment. We talk about nutrition having an impact on phenotypes. Gene expression is turning on and turning off specific genes to produce those specific phenotypes that are desired in different cells and different tissues and different organisms at different points in a lifetime. So our gene expression can change as we age, which is pretty cool. Genome, again, is the sum of all of our genes. Transcriptome is the sum of all the messenger RNA strands that we produce through transcription because we have that genome. And then the proteome is the sum of all the proteins that we make through translation, given the messenger RNAs of our transcriptome. So again, we call it central dogma. All these things are related. We have our DNA. We use our DNA through transcription to produce some messenger RNA. That messenger RNA is used in translation by those ribosomes to make those polypeptide chains that eventually are modified into functional proteins. One way that we can control which proteins are being made by a cell is through the regulation of transcription, choosing which pieces of DNA are being transcribed into messenger RNA. We can shut down transcription by adding DNA binding proteins, which are literally proteins that bind to DNA, right? So what's kind of cool about DNA, that double helix results in a couple different shapes and sizes of grooves. And we have major grooves that are a little bit bigger and minor grooves that are a little bit smaller. And some DNA binding proteins will sit in those minor grooves. More actually will sit in the major grooves, just because, I don't know, they're bigger, I guess. One of the regions where these DNA binding proteins really like to sit is what's called a data box. And it's literally thymine, adenine, and then maybe a couple more adenines thrown in there. And these DNA binding proteins will come and sit here. And if we have a DNA binding protein here, which is part of the promoter region, then what happens is nothing. So our transcription factors are not able to bind and make things happen. The RNA polymerase is not able to bind to the promoter and then initiate that transcription. So these binding proteins will literally block that RNA polymerase from getting to the gene. But we could also enhance transcription by using, you guessed it, an enhancer. So these guys, if we bind some proteins here, we have extra transcription happening. We have another enhancer here. We also have a silencer, which is kind of like that DNA binding protein shutting down the promoter. So lots of ways to make more transcription happen, some ways to completely prevent transcription from happening, and this is all going to lead to more or less transcription. And if I have more messenger RNA, I can make more protein. And if I have no messenger RNA, we're making no proteins. Another way that we can regulate transcription or engage in some selective transcription is through the use of operons. So operons are pretty cool. We have this region of genes that code for some protein or protein complex. Upstream of those genes, we have some other genes. And one of those other genes makes a protein that is a repressor protein. That repressor protein binds to the operator of the promoter region and literally represses the ability of RNA polymerase to sit on the promoter region and transcribe these genes that are downstream of the promoter. But when an inducer comes along, and in this case, the lac operon, the inducer is lactose because these genes are coding for lactase. And in bacteria, why would we make lactase if there's no lactose available for digestion? But when lactose is in the environment and we could use it as a food, we want to go ahead and make that lactase. So this lactose is an inducer and it binds to the repressor and it pulls that repressor molecule off of the operator, which means that polymerase can come in here and code these genes, make some messenger RNA. That messenger RNA can then be used in translation to make some functional lactase, and then this bacteria can use that lactose as a food source. When lactose is gone, those repressor proteins will again bind to the promoter and the bacteria cell will stop making lactase. So super cool way to turn on and turn off some genes. This is called the lac operon, which controls the production of lactase. There are other kinds of operons that do other things, but this one's a super cool one, I think. Another way that we can regulate transcription is through the use of hormones. Hormones are chemical signals produced by endocrine glands. They are transported through the circulatory system and eventually run into some target cells. In this instance, we're looking at the molecule estrogen. Estrogen is a hormone that is sometimes spelled with a silent O, so just be aware that both of those spellings are correct for estrogen. Estrogen is a lipid molecule. It is cholesterol based, and that makes it nonpolar. Because it's nonpolar, it can transport pretty easily right through the cell membranes, and then on the insides of our cells, they bind to some estrogen receptors. We now have an estrogen receptor complex. This can then diffuse into the nucleus. It engages with some transcription factors, and then transcription is activated, stimulated, and we get some messenger RNA being produced. In this instance, we are looking at how estrogen can code for the proteins that eventually help us make some progesterone, which is another hormone that does other magical things. A super cool extra little side bit about estrogen, it can also bind to some cell membrane protein receptors and signal some kinase production, so that cell signaling cascade, which also can lead to transcription. Estrogen also can engage with mitochondria and change the activity of mitochondria in our cells. So all kinds of super effects taken care of by these hormones, estrogens. Birds also make estrogen. Here we're looking at a particular type of estrogen that actually helps birds code for the production of this protein called vitellogenin. So vitellogenin is one of the proteins found in egg yolk. And if a bird is trying to lay some eggs, it needs to make some egg yolk proteins, this vitellogenin. So the estrogen is going to promote the transcription of the vitellogenin gene, which makes the messenger RNA, which then codes for that protein. What's really amazing is that the bird doesn't want to have to waste a bunch of energy producing that messenger RNA over and over and over again to make lots of that messenger RNA, to make lots of that protein. But instead, we're going to code for the messenger RNA a little bit, but then this messenger RNA is going to hang out in the cell and get used over and over and over again. So one messenger RNA transcript can lead to lots of protein production. This is due to the stability of this messenger RNA. But sometimes we don't want our messenger RNA to hang around a long time, and so we can degrade the messenger RNA more quickly if we want to. So another way that we can regulate protein production, here we're specifically regulating translation, is by altering the rate at which we are breaking down our messenger RNA. So here we have a piece of messenger RNA. It's got its 5' cap and its 3' poly A tail. That poly A tail gets chopped up by this enzyme, literally chops off A's, 1A at a time. The longer that poly A tail, the longer it's going to take this guy to chop through all those A's before it gets to the actual part of the messenger RNA that codes for the protein. So if I slow down how fast this enzyme is able to get to these pieces of the messenger RNA, then I can let that messenger RNA sit in my cell for a longer amount of time. What's pretty cool, we could chop off like 30 of these A's every single minute, and so maybe this messenger RNA sits in a cell for just 5 minutes, right? And it's going to code for a protein or two, maybe. But we could also have pieces of messenger RNA where we're taking off only 1A every hour or so. And so our messenger RNA could hang around for a week or so, making lots and lots of protein. And so again, we can edit the rate at which our messenger RNA is being broken down. We can chop it up super fast, and then it's only making a little bit of protein. We can slow down how fast our messenger RNA is getting degraded, and then that messenger RNA transcript can make lots of protein. So again, we're affecting how much gene expression, how much protein is getting produced by changing the rate at which we are degrading our messenger RNA. But wait, there's more. In addition to those ways that we regulate which pieces of the DNA are being transcribed and how long our messenger RNA can hang around in the cell to produce proteins, we also have this super cool thing called epigenesis. And this is super important for the differentiation of our cells, especially in those early embryonic stages. So we've got these cells, these super cool stem cells, and our genome is present in all of those stem cells. Our epigenetic regulation, we're going to talk about these super cool tags that we add to our DNA and to the histone proteins that our DNA wraps around. These epigenetic tags affect the genome, activate some genes, deactivate others, and that leads to some cells turning on some genes and not others. Having different proteins produced in the cell is going to lead to morphogenesis, the generation or the production of different shapes of cells or structures, and that's our differentiation. That differentiation leads to different tissues and different organs, and then those guys, again, due to epigenesis, that gene expression regulation, we're going to produce more or fewer proteins from our genome. So lots of super cool ways that we can get our cells to differentiate so that we can get all the amazing different tissues and organs that we need to be full and complete organisms. The first of the epigenetic tags that we're going to look at, epigenetic tag being those chemical groups that we can add to our DNA or the histone proteins that our DNA wraps around, epigenetic tags that turn on and turn off genes. The first one we're going to look at are methyl groups. The addition of a methyl group is methylation. So here we have cytosine, one of our nitrogenous bases, and on carbon number 5, we have a hydrogen group. But we can actually chop off that hydrogen group and substitute what we call a methyl group. So methane is CH4. If I take methane and chop off a hydrogen, I now have something that wants to stick onto other molecules. This is called a methyl group. So methyl group is here, bound to our cytosine. We're now going to call this methylated cytosine or 5-methyl cytosine because we've got a methyl group on carbon number 5. What's super cool about these methyl groups is that it prevents RNA polymerase from being able to bind to the promoter of a gene. And if I cannot get polymerase on my promoter, we're not going to have any transcription occur. But if we demethylate the promoter, then it is now unmethylated. There's nothing preventing our polymerase from getting there. Polymerase will get there. We will transcribe the gene. We get the messenger RNA that can be translated, and then we have our functional protein. So what's interesting is this is not a mutation. This is not we're changing our ATCG sequence. It is simply a tag that we can add to the DNA that edits whether the gene is going to be transcribed or not. So it does not have effect on the genome, no effect on the genome. Ooh, I didn't spell that correctly. But it does have an effect on the transcriptome, and it does have an effect on the proteome. What messenger RNA is being transcribed and what proteins are being translated. What's really amazing is that this methylation, demethylation changes over an organism's lifetime. And so when we're young, we make some genes, but then when we're older, we don't want to make those genes anymore. And so we can demethylate or methylate or demethylate again the genes that we want to turn on and turn off. Sometimes those methyl groups bind to the actual DNA, to the cytosine of the DNA, but sometimes they'll instead bind to some of the histone tails. So here we're looking at a computer-generated model of a nucleosome. Nucleosome, more vocab reminders. This is the DNA wrapped around its octet. Octet is 8 of histone proteins. So here's our DNA, and then here are those, oh, let me get a better color. Here are those 8 pieces of protein, that octet of histones. What's pretty cool is that some of those histone proteins have these super cool tails that stick out. We can also add methyl groups to those tails. So we can methylate the histone proteins as well as the DNA itself. This is also going to shut down our transcription because the methyl groups on the histone tails cause the nucleosomes to condense. This is also one of the ways that we get our chromatin to condense into chromosomes during prophase of both mitosis and meiosis, adding those methyl groups. But what if I actually want to get to the genes that are here on my DNA? What we can do is add, instead of a methyl group, an acetyl group. So acetylation, the addition of acetyl groups actually opens up the DNA, causes those nucleosomes to spread apart, and it's way easier for RNA polymerase and the transcription factors to get to the gene. So we can open up or add access to the genes by adding acetyl groups. This is how I remember it. Acetyl groups add access, so we can actually do some transcription. The methyl groups mute the genes. No transcription is occurring. There's one more super cool thing that we could add. We could also add some phosphate groups. So we could add a phosphate group instead of an acetyl group. Those phosphate groups are added to our nucleosomes when there's some kind of breakage in the DNA. So lots of phosphate groups will be added if there is something wrong with our DNA, and that is going to promote some DNA repair. I remember that. Phosphate for fix. Good. Hopefully that helps. Those little mnemonic devices help. So methyl mute, acetyl access, phosphate fix. Good. All right. One of the coolest ways that we study epigenetic tags, these methyl groups, these acetyl groups, is by looking at twins, identical twins. So monozygotic twins, these are identical twins. There was a single egg and a single sperm that formed a single monozygote, that first fertilized cell. Sometimes through mitosis, we end up with a big ball of cells. Sometimes that big ball of cells will split into 2 balls of cells, and then those 2 balls of cells will each develop into its own individual. But because they came from the same egg and the same sperm cell, we have genetically identical individuals, hence identical twins. Twins that are fraternal, separate eggs, separate sperms. So not so identical. What's pretty cool when we look at the methylation patterns of very young identical twins, monozygotic twins, we see some pretty similar epigenetic tags. So here we have methyl patterns, methylation, in pretty much the same spots between these chromosomes from different twins. But when I look at the chromosomes of older twins, twins who have lived separate lives, different environments, different experiences, we see where we have different epigenetic tags, methyl groups in different places on those older twins. So super cool how these methyl patterns will change as we age. We're going to turn on and turn off different genes, but also our environments are going to affect which methyl patterns we get, and different experiences are going to lead to different methyl patterns. What's pretty cool about those epigenetic tags, those methyl groups, the acetyl groups, is that they can actually be passed on through mitosis. And so here we see our methyl groups tagged onto our DNA, and then we're coding for our new strands of DNA. And that guy's got the methyl tag, but oh, it's not over here yet. Luckily, we have a protein called DNA methyltransferase that will go ahead and copy the methyl groups onto both daughter strands of the DNA during mitosis. This also happens during meiosis, and so eggs and, well, that's the egg and sperm cells will also have their own epigenetic tags, their own methylation and acetylation patterns. And so that first zygote, the fertilized egg cell, is going to have those epigenetic patterns from both parents. But then we start to demethylate, first the paternal nuclei and then the maternal nuclei, and then we end up with kind of a fresh slate for that embryo. Only specific methylation patterns are going to be reproduced or maintained. And this works for about 99% of our methylation patterns, but there are some things that actually can be inherited. There have been some very interesting studies done. Parents who lived through some severe hunger, so starvation environment, produce specific proteins that help them cope with the trauma of starvation. The embryo, the infant, the new human, may not actually be living in starvation still, but is going to make those same proteins that help the organism cope with that trauma, even though they're not living through that trauma. And so environmental impacts on parents can absolutely sometimes, every once in a while, be passed on to their children, which is weird and interesting. But that's not quite as weird as the story of Ligers and Tigons. So lions, when lions breed, when they make babies, they are pretty polygamous. And so a female lion will mate with lots of male lions. And so her cubs, that litter, could have multiple dads. The dad lions are like, I want my cub to be the biggest cub so that it can survive the best. My genes get passed on. And so they turn on these genes for more growth. The female lionesses, they want to have as many babies as possible. A bigger litter is better for their genes to get passed on. And so they turn off the grow big genes because they don't want a couple big cubs, they want a lot of cubs. So the male lion has these grow big genes turned on, and the female has no, don't grow big. Her genes are turned off. But tigers are different. So tigers have only one mate per breeding season. And so both of them are invested in the growth of their offspring. And so both of them have, yeah, grow, but don't grow that big, but you could grow, it'll be good. So tigers have on genes, but not so big on genes like the male lions. If we mate a male lion and a female tiger, we get what's called a liger. That liger has the epigenetic tag from the dad lion that says grow big, and also the epigenetic tag from the female tiger mom that says, yeah, go ahead and grow big. And so ligers are enormous creatures. That's different from if you have a dad tiger and a mom lion. Over here we had the dad lion and the mom was the tiger. Now we have the dad is the tiger and the mom is the lion. Dad's epigenetic tag that says go ahead and grow gets passed on to its offspring, but mom's lion, no, don't grow that big, also gets passed on. So these epigenetic tags get passed on to this offspring, which is not a liger, it's a tigon. And they're not that big because tiger's little on gene and mom's off gene code for something that's a little bit smaller. Crazy, cool, and interesting. So this epigenome, I'm going to throw another vocab word into our list of vocab. The epigenome is the sum of all the epigenetic tags that can get passed on. Genome, all our genes. Transcriptome, all of our messenger RNA. Proteome, all of our proteins. Epigenome, all of those tags, methylation, acetylation patterns that can also sometimes be inherited. A few slides ago, I don't know, maybe like 8 slides ago, we were talking about estrogen, which can be spelled with an O, and how it affects transcription. So it's a chemical signal in the environment of the cell. It can diffuse into the cell, do some super cool things, and then we have more transcription. We make more progesterone. We make more vitellogenin if we are birds. What's interesting is that chemical signals from the environment can also affect epigenetic tags like methyl groups. A study was done in China, lots of people living with lots of air pollution in the cities, people living with less air pollution outside of the cities, and their methylation patterns were analyzed. And it turns out that people exposed to more long-term air pollution have lower methylation. So less methyl groups if you're exposed to more air pollution. What's important about that is that those methyl groups not being there are going to reduce that transcriptional regulation, which is going to lead to the production of some proteins that cause our mitochondria to work less well. Less functional mitochondria means less good ATP production. That's not good. And then it also can have some impacts on the proteins of our immune system. And so people are getting sick, not just because air pollution is sticking to their lungs, but also because the air pollution is affecting the methylation patterns, which is going to then affect gene transcription and translation and lots of other things. What's worrisome about this is, what did we just talk about? Sometimes those epigenetic tags can be inherited. And so it could be that these people exposed to air pollution, even if they move out to the country and have children where the air is clean, their children could still have some mitochondrial dysfunction and immune response if these epigenetic tags happen to be inherited. And on that note, my friends, we made it to the end of our video lecture on gene expression. We talked about that vocab. We talked about the regulation of transcription, those DNA binding proteins that literally bind to DNA, either in the minor groove or the major groove. TATA boxes in the promoter region. If the DNA binding proteins block those TATA boxes, no transcription happens. If they bind to enhancers, more transcription. If they bind to silencers, no transcription. Operons are clusters of genes that have upstream promoter regions, and the repressor proteins can bind to the operators and shut down transcription. The inducer molecules can bind to the repressors and pull them off of the operators so that transcription can occur. So we can turn on and turn off genes pretty quickly depending on environmental factors. We also talked about hormones like estrogen and how it can induce transcription. Translation, the production of proteins. We can affect how much protein we're producing from a single piece of messenger RNA by changing the rate at which it gets degraded, and that poly-AHL is super important to that degradation. Epigenesis is adding some super cool tags to our DNA and our histone proteins. We have methyl groups, those mute or turn off some genes. Acetyl groups add more access, turn on some genes. Phosphorylation, phosphate groups help to fix DNA when there's some breakage, promote some repair. We talked about how some of those epigenetic tags can actually be inherited. And we also talked about how the environment can affect our epigenome, that collection of epigenetic tags that we have. Great work today.